Have you ever wondered how your body gets energy from the food you eat? It's an amazing process, and a tiny molecule called insulin is the superstar of the show. Imagine you just enjoyed a delicious meal, maybe some pasta, or a piece of fruit. Your body breaks that food down into a simple sugar called glucose. This glucose travels through your bloodstream, ready to power up all your cells. But there's a catch. Your cells are like locked houses, and glucose can't just barge in. It needs a special key to unlock the door, and that key is insulin. It's a hormone, which is a chemical messenger, made by a very cool organ called the pancreas. Think of insulin as the traffic controller for sugar in your body. When glucose levels in your blood rise after a meal, your pancreas gets a signal to release insulin into the bloodstream. This tiny but mighty molecule then travels all over your body, carrying a very important message for your cells. Its job is to tell your muscle, fat, and liver cells that it's time to take in that glucose for energy or to store it for later. So, insulin is the key. But every key needs a lock, right? In our bodies, that lock is called the insulin receptor. It's a big, complex protein on the outer surface of your cells, sticking out like an antenna, waiting for a signal. Think of it as a special docking station just for insulin. These receptors are found on many different types of cells, especially muscle cells, fat, adipose cells, liver cells. Each receptor is made of 4 parts, 2 on the outside, the alpha subunits, 2 that span the membrane, the beta subunits. These parts all work together as a team. The 2 alpha subunits on the outside are the landing pads for insulin. They're perfectly shaped to grab an insulin molecule as it floats by in the bloodstream. When insulin isn't around, the receptor is just chilling, off or inactive. Other hormones and molecules float around in your blood, but the insulin receptor ignores them. It's exclusively designed to bind insulin, delivering the right message to the right place at the right time. Now, let's get to the really cool part, the shape shifting. When an insulin molecule, our key, finds its receptor and binds the alpha subunits, it's like putting the key into the lock. That binding causes a physical change in the entire receptor structure. The alpha units pull closer together, which pushes the beta subunits inside the cell closer to each other. It's like a molecular hug. This change in shape wakes up the part of the receptor that's inside the cell. The receptor goes from off to on, a physical trigger that starts a chemical reaction. The beta subunits, now pushed together, have a special ability. They are enzymes. They can kickstart chemical reactions. Specifically, they are tyrosine kinases. They take a phosphate group, a tiny charged particle, from ATP, the cell's energy currency, and stick that phosphate onto specific parts of themselves and other proteins. This process is called phosphorylation. The first thing the activated receptor does is add phosphates to its own beta subunits. It's like the receptor high-fiving itself to celebrate receiving the insulin signal. Once the insulin receptor has activated itself by adding those phosphate groups, it's ready to pass the message along. This is where the chain reaction, or signaling cascade, truly begins. Think of it like a line of dominoes set up inside your cell. The activated receptor is the finger that pushes over the first domino. This first domino is a group of special proteins waiting just inside the cell membrane. They are called insulin receptor substrate proteins, or IRS for short. The activated receptor uses its kinase power adding phosphate groups to the IRS proteins. The IRS proteins are now tagged with phosphates. Those phosphates act as docking sites for other proteins, like a sticky spot on the first domino that the next domino connects to. One important protein that binds phosphorylated IRS is PI3 kinase. PI3 kinase docks onto IRS and becomes activated. The signal passes from protein to protein through activations. Each step amplifies the signal, making sure the cell gets the message loud and clear. Insulin has arrived. PI3 kinase's job: work at the membrane, create special signaling lipids. Those lipids recruit another crucial protein to the membrane. That next major player is ACT, also called protein kinase B. ACT is brought to the membrane, then phosphorylated and switched on. ACT acts as a master switch. It can travel and turn on or off many processes. ACT activation is a pivotal moment. The message splits into several effects. It tells the cell to start making glycogen, a storage form of glucose, to build new proteins to grow and survive. Most importantly, ACT triggers the final step to get glucose into the cell. So, the master switch, ACT, is now on. What's its most famous command? It tells the cell to move its glucose transporters to the surface. Let's break that down. Inside the cell, there are little storage bubbles called vesicles. Tucked away inside these vesicles are special proteins called GLUT4 transporters. Think of GLUT4 as the actual gateways or doors for glucose. Most of the time, these GLUT4 doors are kept hidden away inside the cell. The cell is essentially keeping its doors in the garage until they are needed. When ACT is activated, it sends the signal to these vesicles. The message is simple: move to the surface. Vesicles travel to the cell edge. They fuse with the outer cell membrane. As they merge, the GLUT4 transporters become embedded in the cell surface, facing outward. Suddenly, the cell that was a locked fortress now has dozens of open doorways for glucose. With the GLUT4 gates open, glucose from the bloodstream can finally enter. Glucose moves from high concentration, blood, to low, inside the cell. This is facilitated diffusion. It doesn't even require energy. GLUT4 simply provides a channel for glucose to flow down its concentration gradient. The cell can now use this glucose immediately for energy. In liver cells, glucose molecules are linked to form glycogen for storage. In muscle cells, glucose is also stored as glycogen for later use. Mission accomplished. This pathway is fundamental to our health. It's how our bodies regulate blood sugar and keep cells fueled. When it works well, we have energy and normal function. But if the key, insulin, isn't made, or the lock, the receptor, is broken, or the receptor becomes resistant to the key, the whole process fails. This can lead to high blood sugar and diseases like diabetes. Understanding this molecular machinery shows how elegantly our bodies are designed, and why keeping this pathway healthy matters. Consider the science. So what is this insulin really? It's a protein, a complex molecule built from smaller pieces called amino acids, all folded into a specific shape. Its unique shape is what allows it to function as a perfect key. It's designed by your body with incredible precision to fit into a very specific lock. This lock isn't on a door, but on the surface of your cells. The interaction between the insulin key and its cellular lock is the first step in a fantastic biological chain reaction. This process happens billions of times a day in your body, all without you ever having to think about it.